Transduction of the ear by Gabby Jones. The ear is responsible for translating variations in air pressure into the neural activity necessary for our idea of sound. The ear is divided into three sections: the external ear, middle ear, and inner ear. The auditory meatus of the external ear gathers sound energy and focuses it on the eardrum or tympanic membrane. From the ear canal, the sound wave then vibrates the eardrum, which vibrates the three tiny bones of the middle ear known as the malleus, incus, and stapes. Stapes vibrates a small membrane at the base of the cochlea known as the oval window. This sends amplified vibration energy to the fluids of the cochlea. The inner ear converts sound into neural activity. The part of the inner ear that is responsible for the auditory portion is the cochlea. It is made up of the base and apex. Within the cochlea are three parallel canals: the scala tympani, vestibuli, and media. However, the main structure for converting sound into neural activity are found in the basilar membrane. The basilar membrane vibrates in response to sound transmitted to the fluid-filled cochlea by the oval window from the bones of the middle ear. Within the scala media and basilar membrane is the organ of Corti. The organ of Corti contains sensory hair cells, supporting cells, and the ends of auditory nerve fibers. Human ears contain one row of inner hair cells and 3 rows of outer hair cells. The inner hair cells and nerve fibers help with the perception of sound. The outer hair cells receive inputs from the brain that sharpen the frequency power of the cochlea. The upper end of each hair cell contains stiff, tiny hairs called stereocilia. On top of the organ of Corti is the tectorial membrane. The stereocilia extends into the tectorial membrane. The movement of fluid in the cochlea produces vibrations of the basilar membrane. This bends the stereocilia inserted into the tectorial membrane. Depending on the direction of the bend, ion channels in the hair cells open or close. A change in ion conductance in the hair cells will increase or decrease the firing rate of the auditory nerve fibers. The tip links connect the stereocilia. They produce the means for rapidly translating hair bundle movement into receptor potential. Opening of the channels will cause an intake of potassium ions and rapid depolarization of the entire hair cell. The depolarization leads to an increase in calcium ions through voltage-gated calcium channels at the base of the hair cell. The calcium triggers the release of neurotransmitters from the synaptic vesicles at the base of the hair cells via exocytosis. This stimulates the nerve fibers, and the signal is then passed to the brain.